All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. I cannot believe that I have now done 15 videos about Novavax. I created a playlist. The link to the playlist is in the description of this video as well. I thought that the Novavax's representation, although as much as there were lighting issues and those, but their representation and their presentation in the FDA meeting was very interesting, especially this part of the message that as the Novavax vaccine has the spike protein with conserved epitopes, meaning parts of the spike protein that are conserved across variants, and their vaccine triggers antibodies against those epitopes as well. That means the vaccine has efficacy across variants. So they had the data for prototype. Prototype is original Wuhan. And before Omicron, they had the data for preclinical data for Omicron as well. Preclinical meaning in vitro studies, just like New England Journal of Medicine, where we presented the Harvard study, which was in vitro study as well. And they showed that the Novavax vaccine efficacy against even Omicrons was fine. So we have a broken science system in general. This is just one comment, and then we'll go and look at the data. We have a broken system, and that system is broken in this way that there are studies like from Harvard that say the current vaccines, current infection protection is useless against the newer variants. And there are studies which show that the current vaccines and variant vaccines are working fine. Then there are studies that in vivo, in people, show vaccines are not efficacious. So I think you can tell that there is a broken science going on at this time. And I intend to do a discussion about that later on. Let's look at the Novavax, which I was impressed with. And I do not have any shares or interest with Novavax. So this is not a commercial selling of Novavax. I just think Novavax is an interesting vaccine for COVID. So this is drbeen.com. In the description, there is a link to another 900 lectures at the price of $67, one-time fee. I don't think you could ever get access to 900 lectures. Unlimited access, plus any new lecture that comes in is also included. However, if you're watching this in the future, maybe a month or so later, we are not going to be offering this anymore. 67 is actually ridiculous. You cannot give even one video for 67. So if you click it after some time and you are not able to, my apologies. Okay, now let's look at the links. This is World Health Organization on 13 June. We'll look at that a little bit. This is the Novavax, and thankfully they have corrected their links so they do not have to be downloaded anymore. This is the 28 June. Novavax to participate in vaccine. So they were very happy about this, that they were participating in the, or they were invited in the FDA committee meeting, which means they, at least from within the work, within the processing of that machine, working of that machine, they are accepting Novavax as a candidate. So they were happy about that, and this is that news. Then here is Novavax COVID data presented at FDA. So this is their summary of the data that they presented to FDA, and then we actually have that presentation here as well. So I want to first go over the summary, and then we'll look at the presentation a little more in detail. In general, there is good news that Novavax looks good. So just give me 1 second. My apologies, my layer. Okay, so Gifts for Humanity, they continue. So how did Novavax fare during the FDA Advisory Committee meeting? So this meeting that was yesterday, I think it was for the booster for Omicron in fall. Was FDA, CDC, are they going to offer a booster? And then the question really that is burning in my head is, are they going to say, yes, we are offering a booster, and then they say we are mandating the booster for healthcare workers and others? I think that if you look at the data, it does not look like, at least from the data that I presented yesterday, so I'm not going to talk about it again. I don't know if from that data it seems like a booster is urgently necessary. So let's see. Their vaccine is Novavax vaccine, NVX CoV 2373. And according to them, these are their statements. They cover Omicron and other circulating variants. And I'll show you their presentation. I loved it. Preclinical data. So the data before, so non trials that are not in humans, in vitro trials. Boosting with Novavax, Omicron or prototype. So here, Omicron vaccine is the one that is specifically targeted at the spike protein of the Omicron species. I'm calling it species 1, 2, 4, 5. And prototype is the Wuhan. It's not alpha, it's not beta, it's not delta, it is Wuhan. So either we have Wuhan or we will have Omicron. So they're saying we use Omicron only, or we use bivalent, that is prototype plus Omicron, or only the prototype. In all cases, their vaccine shows good efficacy, and they have a reason for that, and we'll see that. They also say that Novavax is ready to supply Omicron-based vaccine this fall. I remember the question was asked from them that, hey, are you ready? So then they said the question was a little less definitive, the answer. But anyways, on their website, they say that we'll be ready by fall. We'll be ready to provide the doses by fall. Now, why is it that their vaccine covers everything? Here is a simple answer from them. This is their presentation. We'll go over that later on. What they're saying is that they are using a spike protein. Of course, we know that. And their spike proteins are in the form of trimers. That means that there are three lines, three spike proteins together, and then they put that in a fruit basket bouquet like thing, and then they have such trimers, and they make a particle with that, and that is what they call nanoparticle. This nanoparticle is not messenger RNA based nanoparticle, so it has nothing to do with that. It just is a small collection of spike proteins. That is why it is called a nanoparticle. The spike protein that they're using has, if you see here, if I can make it a little bigger, if you see these blue little areas, this is the same exact epitope, the same area. It is not mutated, it is not changed. It is the same area that is present in all variants. Because of the mutations, the place may be changing a little, but it is present in all variants. So this is what they're calling a conserved epitope, and they're correct in doing that. So their Novavax vaccine antigen, the spike protein, contains conserved epitopes that are conserved across the viruses. Because of that, when the antibody is formed against this area, this antibody will attack the same epitope everywhere else as well. That is their point. So, good one. And I would suggest that if this is the case, and if others are also, for example, there is messenger RNA, and that messenger RNA makes a spike protein that also has a conserved epitopes, then it should work as well. So once again, we hit against the broken science that on one end, if you look at this, you would say, yeah, sure, messenger RNA or the spike protein, subprotein based vaccine should work fine across. But why is it losing efficacy then? Okay, so continuing. Here, if you see, they said that for our spike protein, when the antibodies are formed, not by us, but these are the monoclonal antibodies that are formed in lab, these antibodies attack this conserved region. Because of that, it doesn't matter it is prototype, that is Wuhan, or Omicron BA1 or BA2 or BA4 or 212.1. 212.1 was dominant in the US at this time. I think from yesterday onwards, BA4 and 5 have collectively become dominant. So once again, Novavax is saying that, hey, these monoclonal antibodies that we have attack this area and that is conserved. So that means when we will get the vaccine and produce similar antibodies that would do the same behavior. Then this is also a very interesting point that they brought up. I really thought that was fun. I think that is because I love medical concepts. So here they said, so some of you are going to throw something at the monitor, but hear me out. This is an interesting medical concept. They said that as more doses are given, so okay, don't throw anything yet. Hear me out. Medical concept. They said as more doses are given, the immunity becomes better. So we know that the general rumor is if you give more doses, you make immunity bad, or more doses and still no efficacy, and so on. So, from a mechanism point of view, what are they trying to say? It's actually cute. What they're saying is the following. Imagine we have various variants. We have, let's say, we start from this green thing over here, that is Wuhan, and the variants that are near this. Near means more closely, more similar to this. Then we have alpha that is a little distant, delta is distant, beta is distant, and here BA5 is too distant in terms of the antigens. So what they're trying to explain here is if you're making antibodies here, then these antibodies may not be as effective for here. However, note this, they are saying that we are giving you the spike protein, and that spike protein has epitopes that belong to this area. So let's say, I'm going to make it simple. Let's say this epitope here, or let's make it on the RBD side. Let's say this epitope here is the epitope for Wuhan. That is, it has a conserved area that is also conserved in the Wuhan variant. So when you give this spike protein, and if we make the antibody against this area, then we'll be able to cover the Wuhan variant. But in theory, we may not be able to cover BA5 because it is distant. And so this epitope, let's say BA5 spike protein, has this epitope, but that is kind of little mutated. So this antibody that we make will not attack here very well to BA5. But the question to answer is, so keep an eye on this one. How could you continue to give the same one spike and still produce adjacency to the distant ones? Here is how. Imagine that there is another epitope here, which is on the Wuhan and is also on the BA5. So once you give the vaccine, imagine that with the first two doses, body made predominantly the antibodies against the Wuhan type. Maybe it also made it against this one, but let's just assume mostly with the Wuhan. Then you gave a booster, and then body made maybe some antibodies against this other epitope. And now the vaccine coverage of a more distant cousin has become better. And then you give one more booster. So I know that you're going to be upset with me about boosters, but mechanism, let's just keep the mechanism in our mind. So if you give one more time the booster, then you expose the body to that spike protein once more and allow it to create more antibodies even to more areas, which would continue to create more and more similar areas, or create antibodies against more and more conserved areas that are conserved across these spike proteins. So this was a very cute way of putting it together to say, as you're getting boosters, the distance between the variants in terms of coverage become less and less. So I thought that is, and it's a valid phenomena. I can give you a counter to this phenomena. The counter to this phenomena is I'll actually give you two counters. One counter to this phenomena is that what if this does not happen? What if the body decides not to make antibodies against the other areas? It's not necessary that with each booster we will end up here. That's one counter. The second counter is what if you keep boosting and the body is continuing to attack the previous epitope because of original antigenic sin? And that would actually cause more side effects but less advantage. Now, the mechanism that I mentioned, the counters, are more hypothetical, but they are possible. So that means it's not necessary that what they are saying here will happen. However, this is a mechanism to keep in mind as well. It's a very interesting mechanism. So they are saying that as you boost more, there, I would also actually say this, which part of the antigen our body attacks with the first dose or the second or the third or the fourth is actually not under anybody's control. It is not this that third dose will actually do better or fourth. Maybe with the very first dose or second dose, our body would start attacking the right epitopes and will be fine. So this behavior is random. The only thing is what they're saying is if you keep exposing the body to the antigen, then the body would continue to have a chance to make more and more antibodies against other epitopes on this antigen. So is it going to be always the case? Don't know. I would disagree, but still interesting mechanism. Now with this, we are done with the basic discussion. Summary is done and some interesting points are done. So if you just wanted to stop here, you can. I want to show the presentation itself. It's a short presentation, not too large, but still interesting. And by the way, Novavax's stock has been rising. So if I go to see Novavax stock, it was dropping. So if you see here, this was what? If I go to, let's say, 6 months, they had really gone down to even 38 here. This is June 14. So 28 today, 14 days ago. And so they have risen back to 51, 52. Again, I don't have any commercial interest, so this is just interesting. Okay, so now Novavax presentation. I'm not going to go through the whole thing. I just want to make sure we know that this is a recombinant protein, subprotein vaccine. That is, they have made these little tiny spike proteins and they have attached them to a thing with matrix M adjuvant, and that is a lipid nanoparticle. But this is not the mRNA type lipid nanoparticle. Actually, I shouldn't even call it lipid. It's a nanoparticle. It has matrix M adjuvant with it. I've discussed many times. Here is the efficacy. High levels of protection achieves in two phases, phase 3 trials, actually, two phase 3 trials with Novavax. One is study 302 UK. We discussed that in the past. Study 301 US and Mexico. Overall efficacy, mild, moderate, severe, 90% in both. Severe disease, 100% efficacy. Against variants of interest, various variants. Interest and concern, 86% to 93%. Now remember, it is for 18 years and older. Symptomatic and asymptomatic illness through 6 months, 83% and 71%. So decline or waning is not as fast. Then you saw this one that they say that our protein contains conserved epitopes. Then you saw this one as well, that their monoclonal antibodies, when they have, these are against those conserved epitopes. Then clinical evidence of enhanced breadth of antibody response. So that was the important discussion yesterday, that can it cover the current vaccine? Can it cover Omicrons or not? Or do we need to make a new vaccine? And do we need to mix that vaccine with the older ones? And I had that discussion yesterday, so I'm not going to do that discussion again. But here is their presentation of saying how their vaccine is able to work with it. So here, study 301, immunoglobulin G associated with COP research reaches high levels after boosting variant responses. So if you see here, the green one, is prototype. Then BA1, BA2, BA5, this is day 0 to 21. These are the antibodies. And then here is the 8-month boost. So boosting after 8 months. And if you see here, protection against all variants went up. So that's interesting. Then here, this is ACE2 receptor binding inhibition, so neutralization. Antibodies reach high levels. So again, if you can see here, this is the original, then 8-month boost, and then there is efficacy there. And if you see here, BA4, BA5, sorry, not BA4, BA2, BA5, and BA1, they're all covered. Then here, study 301, UK, US, Mexico, ACE2 receptor binding neutralization. So if you see here, history of SARS-CoV-2 infection, somebody who was infected, this is their antibodies. And then when they boost with this, then here is the outcome. This is interesting. This is that concept that I just discussed. They say that as more boosters are given, the coverage continues to become better and better. And here is the slide that I just showed you, that as the boosters are given, the coverage distance continues to become shorter. Then rhesus macaque, so this is now preclinical, boosting with either prototype. This is very important actually for yesterday's discussion. Either prototype or Omicron. I would have wanted just one type instead of mixing both. So here they're saying it, either prototype, that is the vaccine against Wuhan, or Omicron, drives antibody response to more universal-like profile. So they're saying that as you keep boosting, it becomes more and more universal. So that's a beautiful mechanism. So here, if you see, this is Novavax against Wuhan, and then this BA1, and this is bivalent, that is both of them. If you see here, bivalent, that is 2 Wuhan plus Omicron, or this is Omicron alone, they almost have similar behavior. So in my opinion, taking a risk of combining them, I discussed that yesterday, is not necessary. So their summary was high level of efficacy in 2 phase 3 trials. And then Novavax technology induces cross-reactive immunity, so Omicrons and others, driven by recognition of conserved epitopes on Novavax recombinant spike protein. And adjuvant enhances breadth and duration of immune response, so they can go on for 8 months, and the adjuvant causes the immune system to be kicked and kind of react more. Broadly, cross-reactive immunity increases with boosting. This is where they're selling a booster, they're giving a mechanism there as well, but I personally am not very comfortable with boosters. Some people ask me, did I get the booster? I didn't get a booster. Broadly, and that also because I got booster was due, I didn't take it, got the infection. After that, booster was just generally not necessary, at least for me. You make your decision. Broadly cross-reactive immunity increases with boosting. Individuals may prefer prototype-specific vaccine based on known efficacy, extensive safety data. So they're saying that if you wanted to have a boosting, they're showing that the boost with the original one, just the prototype, is working fine. So you may not want to take, let's say, Omicron only, because that may be a new product. So you already know what the previous one is doing, so maybe just boost with that. Boosting with Omicron, so no advantage with bivalent. This is very important. No advantage with bivalent. In my opinion, when I discussed this yesterday, no advantage with bivalent actually means a risk with bivalent, because you have one vaccine, one type that is known to produce a risk, and now that is not being effective, then why to continue to add that risk in there? I did that discussion yesterday. Boosting study ongoing and Omicron specific vaccine available Q4. This is the discussion. I thought it was interesting. So with this, Do me a favor. So Michael says, awesome video and information. Thank you, Dr. Bean. You're very welcome. In the description of this video, there is a playlist, link to the playlist for all Novavax videos. I have individually put the links for each video separately as well. And then there are links in the description for other things too. For example, if you wanted to buy Dr. Bean package, or if you wanted to support this work, there's a link to buy me a coffee, or use PayPal to support this work, or become Patreon member, or member of YouTube, and so on. So with this, thank you very much. I would see you tomorrow. I hope Alquin liked the turtle names. If not, I am we are sorry, but if you did, we're happy. And I would see you tomorrow. Tomorrow we'll do 2 lectures. We'll do this one, plus we'll do art and cafe. Day after, we'll do the questions that we had collected last week, and we'll go from there. Thank you very much. See you tomorrow.